Assalamu alaikum warahmatullahi wabarakatuh. Dear student, today inshallah we're going to talk about the muscular tissue. What is the general structure of the muscle tissue? The general characteristic of the muscular tissue. The muscle is formed of many fibers. These fibers are elongated cells. Each cell has large number of contractile filaments known as actin and myosin. Types of muscles. We have three types of muscles. Cardiac muscle, which present in the myocardium of the heart. It is striated and involuntary. What the meaning of striated? Have lines like this, striation. And the action is involuntary, not with will. I cannot control the contraction. Skeletal muscle are striated and voluntary. I can control the action of this muscle. Smooth muscle is non-striated and involuntary. Here is the skeletal muscle. Skeletal muscle has regular striation and has cylindrical in shape with multiple peripheral oval nucleus. Skeletal muscle, striated and voluntary in action, they appear cylindrical, regular striation, multiple peripheral oval nuclei. The cardiac muscle has irregular striation and the action is involuntary. Has central oval nucleus. The smooth muscle has no striation, spindle in shape, and the nucleus is central, single, and oval, and the action is involuntary. First, we're going to talk about the skeletal muscle. What the name skeletal comes from? Muscles attached to the skeleton. Do all skeletal muscle attached to the skeleton? No. Some muscle skeleton not attached to the skeleton, as that of the tongue, face, pharynx, esophagus, and diaphragm. All the skeletal muscle are voluntary in action except the pharynx, diaphragm, esophagus, and cremasteric muscle. This is a diagram for the skeletal muscle. The whole muscle is covered with a connective tissue known as epimysium. The muscles present in the muscle fiber present in bundles. Between the bundles there is connective tissue known as perimysium. In between the fibers there is endomysium. Again, this is the muscle fiber. Between the fiber there is endomysium. And this is the bundle. And this is the bundle. Between the bundles there is the perimysium. All the bundles covered with epimysium. The skeletal muscle consists of fibers held together and supported by the connective tissue. The epimysium is dense connective tissue surround the whole muscle. The perimysium less dense, and it descends from the epimysium and surround the bundles. And the endomysium is reticular fiber that surround the muscle fiber, each muscle fiber. Again, this is a diagram. This TS in the skeletal muscle, and the connective tissue between the muscle fibers is the endomysium. Between the bundles, this is the perimysium. Surrounding whole muscle is called epimysium. What is the importance of this connective tissue? Force transmission. What do I mean with force transmission? When one contracts, all the muscle fibers contract. Connect the muscle fiber together for strong contraction. It is rich in nerve supply and blood vessels that supply the muscle with nutrition. This is an LM picture of the skeletal muscle. As you can see here, it is cylindrical. This one muscle fiber, this is another. Okay, each muscle fiber is cylindrical in shape. It has a regular striation, transverse striation, and multiple oval peripheral nucleus, and the cytoplasm is acidophilic. Cylindrical in shape, large in diameter, non-branched except in the face and the tongue. The cell membrane is called sarcolemma and it is thick. The nuclei are multiple, oval, and peripheral. The cytoplasm, known as the sarcoplasm, is acidophilic with transverse striation. The smooth endoplasmic reticulum called sarcoplasmic reticulum. This is an LM picture of TS section in skeletal muscle. This section is polyhedral with multiple peripheral oval nuclei. Connective tissue between the muscle fiber is known as endomysium. Between the bundles, there is the perimysium. A very important question: how is the skeletal muscle adapted to its function? This is a diagram showing one muscle fiber. Each muscle fiber contains myofibril, which is the actin and myosin, myofibril. And this is the smooth endoplasmic reticulum. This is the sarcolemma, and this is the sarcolemma. These are the mitochondria. Each muscle fiber has many smooth endoplasmic reticulum play important role for calcium pump, and the calcium pump, as we're going to say now, is very important for muscle contraction, and contain myofibril, which are the contractile filaments. We have numerous mitochondria for energy production. Inclusion as myoglobin, which is oxygen binding protein, and it provides oxygen for the oxidative reaction. Glycogen and lipid found between the myofibril to provide energy. To summarize here, I need the mitochondria, which is the factory for production of energy. I want something to burn, which is whether glycogen or lipid, and I want to burn with oxygen, so I'm having myoglobin, which is an oxygen binding protein. Again, this is an arm. I'm going to have a muscle. From the muscle, these are bundles. I'm going to have a bundle. From the bundle, I'm going to have one muscle fiber, and this is the sarcolemma. I'm going to cut the sarcolemma. I'm going to take one of the myofibrils. As you can see here, the myofibril has striation. This striation due to alternating dark band and light band. Here, the dark band or the A band is formed of actin and myosin. It's formed of actin and myosin. That's why it is dark. And bisecting the dark is the H zone, which is formed only of myosin, then the M line. Here is the I band. It is light as it's formed only of actin, and there is Z line bisecting the I band. The distance between two Z line is called the sarcomere. It is the functional unit for contraction. The EM picture of the myofibril. They are contractile threads. They are longitudinally arranged parallel to the muscle fiber. They are alternating dark A band because it's formed of actin and myosin, light band formed of actin only. The A band has a pale area H zone, which is formed only of the myosin, and a dark line bisecting the H zone. The I band is pale as it's formed of actin only, bisected by a dark line known as the Z line. Again, here is the A bands formed of actin and myosin, the I bands formed only of actin. The Z line bisects the actin. The distance between two Z line is called the sarcomere, and it is the functional unit for contraction. The sarcomere is the distance between two successive Z line. The I band is pale as it's formed of actin. The A band is dark because it's formed of actin and myosin, and the H zone is light as it's formed of myosin only. Here is contraction gliding of actin over myosin. What is the function of calcium, dear students? This is the myosin, and this is the actin, and this is the Z line. Okay? What's going on here? The myosin wants to take the actin inwards in order to do shortening of the muscle. This cannot happen. Why? Because I'm having protein on the binding site that the myosin will bind with the actin with. The calcium will bind with this protein, making this binding site empty in order the myosin binds to it and take the actin inwards. So that's the function of the sarcoplasmic reticulum in pumping the calcium. Here is a diagram showing an important issue here in the skeletal muscle. This is the sarcolemma. Okay, the sarcolemma, what's this? Invagination of the sarcolemma inside the sarcoplasm. And this invagination, as you can see here, the letter T, capital T. Here the invagination occurs between two terminal cisternae of the smooth endoplasmic reticulum, forming what's called triad tubular system. Triad tubular system. Again, invagination of the sarcolemma inside the sarcoplasm between two terminal cisternae of the smooth endoplasmic reticulum. The triad tubular system, by definition, is formed of transverse invagination of the sarcolemma in the sarcoplasm, forming the T-tubule. The smooth endoplasmic reticulum form transverse-wide cisternae known as terminal cisternae on either side of the T-tubule. The two terminal cisternae of the smooth endoplasmic reticulum and the T-tubule in between form the triad tubular system, which play an important role in muscle contraction. While the nerve impulse moves on the surface of the sarcolemma, it will descend in the T-tubule, in the sarcoplasm, stimulating the sarcoplasmic reticulum to release calcium, and the calcium facilitates the sliding of actin over myosin. The second type of a muscle is known as the cardiac muscle, which is present in the myocardium of the heart. As you can see here, it's formed of muscle cells connected together, forming fiber, connected by intercalated disc junction known as intercalated disc. Found in the myocardium, they are striated and involuntary. Why it is irregular striated? Because the muscle fiber branch and anastomose. The fibers, as we said, branch and anastomose, divided into short segments connected together by the intercalated disc. The myocardium is the middle layer of the heart. As we said, it is cylindrical in shape. Intermediate diameter between the skeletal and the smooth muscle. They are branching and anastomose, covered with thin sarcolemma, and the nucleus is single, oval, and central. Again, here is the muscle fiber, the cardiac muscle fiber, branch and anastomose. The cardiac cells are connected together by intercalated disc form the fibers. We having few myofibrils, numerous mitochondria, smooth endoplasmic, Golgi apparatus, glycogen, and myoglobin for energy. What the meaning of lipofuscin pigment? As we said in the H1, the lipofuscin pigment is the waste product from digestion by the lysosome, in which the residual body cannot move away by exocytosis and stay inside the cardiac muscle. And you can see why in the cardiac muscle, because it is not dividing cell. The intercalated disc is junction between two membranes of the cardiac muscle, prevents separation during contraction. What is the function of the cardiac muscle? Exhibit involuntary rhythmic contraction. Smooth muscle, they are present in the wall of the viscera, spindle in shape, with a central single oval nucleus. cytoplasm is pseudophilic. Why it appears non-striated? Because there is an irregular arrangement of the actin and myosin are irregularly arranged. That's why I cannot see striation. Diagram showing smooth muscle. Spindle in shape with central oval single nucleus. Okay, again, dear students, these are different types of muscle. Skeletal muscle in which the striation is regular, multiple peripheral oval nuclei. Here it is cylindrical in shape, okay, intermediate in size between the skeletal and the smooth, irregularly arranged, irregular striation due to branching anastomosis, connected together by intercalated disc. The nucleus is single, central, and oval. Here is the intercalated disc. Smooth muscle, spindle in shape with central oval, single nucleus, and it is non-striated due to irregular arrangement of actin and myosin. This is the table showing you the difference between skeletal, cardiac, and smooth muscle. First, the site, the skeletal muscle attached to the skeleton, while the cardiac present in the heart, smooth muscle present in the viscera. Both the skeletal and the cardiac are cylindrical in shape, while the smooth is spindle. The skeletal is large in size, the cardiac medium, while the smooth is small. The skeletal muscle non-branching as well as the smooth, while the cardiac branching. The thinnest sarcolemma is for the cardiac muscle, then the smooth, then the thick is the skeletal. Striations are clear in the skeletal muscle as the myofibril are regularly arranged, alternating light and dark bands, while the cardiac non-clear due to branching and anastomosing of the muscle fiber, so they are not regularly arranged. While the smooth muscle, absent striation as the actin and myosin are arranged haphazardly, irregular arrangement of the actin and myosin. Intercalated disc present only in the cardiac muscle. The nucleus in the skeletal is multiple peripheral and oval, while that of the cardiac single and central oval, the smooth as well as the cardiac. The action only the skeletal muscle is voluntary, while the cardiac and the smooth are involuntary. Here is light microscopic picture for LS section in the skeletal muscle, cylindrical arranged, pseudophilic, regular transverse striation, multiple peripheral oval nuclei. CS section in the skeletal muscle, polyhedral, the multiple peripheral oval nuclei. This is the cardiac muscle branching and anastomosing. Central single oval nucleus connected by intercalated disc, and this is the lipofuscin pigments. Thank you, dear students.